Hello everyone, welcome back to my channel. In this series of video, I'm discussing blood and its component. In today's topic, we are going to discuss about thrombocytes or platelets. Now, what are thrombocytes? Thrombocytes are small, oval and spherical, non-nucleated structures. Okay. They are fragments of large cells like megakaryocytes. Okay. So actually, thrombocytes are not true cells. They are fragments of large cells like megakaryocytes. Megakaryocytes, where we can find this type of cells? We can find it in red bone marrow cells. So the megakaryocytes are fragmented, okay? And from these fragments, platelets are formed. Now each fragment is covered with a layer of plasma membrane. Fine. So in today's topic, we are going to focus on platelets function. Main function of thrombocytes are blood clotting. Okay. What is blood clotting? Blood clotting is whenever a blood vessel is injured or cut, right? Blood comes out of it. So blood comes out of it in form of liquid state, right? After some times, what happens? Blood changes its forms and form a thick jelly-like material. Now this is called clotting. After this jelly-like material is formed, the blood flowing is stopped. Blood flowing from the injured blood vessel is stopped. So this is what blood clotting is. Okay. Now this helps us in blood loss. If there is some cut, by somehow we got a cut or any of our blood vessel is injured. So if this blood clotting doesn't take place, what will happen? All the blood from our body will flow out and eventually we will die. So this mechanism is very important for us. That is blood clotting, and it is done by thrombocytes. Right. Now, blood clotting is done via few steps. Okay. So one by one, we are going to learn about the steps. Okay. So in blood, there is a protein called fibrinogen. Okay. There is a protein that is present inside blood which is soluble. Okay. Which is soluble in nature. Now, this soluble fibrinogen becomes what? Becomes fibrils of fibrin. Okay. Fibrils of fibrin. Fine. It forms a fine mesh-like structure. It forms a very fine mesh-like structure. Okay. And inside this mesh, other blood cells are trapped. Okay. And this mesh clog the opening. Okay. Opening or wound. Okay. Now this fine mesh of fibril of fibrin protein clog the opening of wound. Right. Now they forms a temporary plug. Right. Now this is how blood coagulation takes place in our body. Now there are some factors which helps fibrinogen to convert into fibrin. Some substances. What are those substances? The substances are prothrombin, prothrombin, an enzyme, thrombokinase, okay, and calcium ions. Fine. So these are the things that are needed for blood coagulation. So step by step we are going to learn about it. Okay. So let me clear this board first. Now step by step we are going to learn. So what is our step number 1? Our step number 1 is whenever there is a cut in our blood vessel, platelets, they release thrombokinase. Okay, thrombokinase. Whenever there is a cut or wound in our blood vessel, platelet releases thrombokinase. Fine. In step 2, with the help of calcium ion, there is a blood protein which is prothrombin, as I have mentioned it earlier, prothrombin. With the help of calcium ion and this thrombokinase, it forms thrombin protein. Okay, thrombin protein. Now this thrombin protein is what? It is enzymatically active. That's why this conversion is needed. Why? Because thrombin protein is enzymatically active. Okay. Now again, fibrinogen, with the help of this enzymatically active thrombin protein, it forms fibrin, insoluble fibrin. Okay, insoluble fibrin. And as we know, this insoluble fibrin is very much important for blood clotting. Right? We will see a diagram and again we will revise this topic. Fine. Okay. There was some delay, but we got the diagram. Okay. So here what we can see. So there is a damaged blood vessel. Fine. A broken vessel. And from here, blood is coming out. These are red blood cells. We have studied that they are biconcave, right? They are biconcave. These are red blood cells. They are coming out from the broken vessel, right? So the clotting factors does what? They convert prothrombin to thrombin, which is an enzymatically active compound. Fine. Thrombin does what? Thrombin convert soluble fibrinogen, which is already present in blood, to fibrin, insoluble fibrin, right? And fibrin forms a fine mesh like structure. You see, these are the fibrin strands. Fine. They form a mesh like structure which stops the blood flow. Hence blood is successfully clotted here. blood is what successfully clotted here and blood loss is stopped. Okay, so this is how blood coagulation takes place. So that's it for today's video. If you like my content, please like, share and subscribe. Thank you.